We're under a little bit more tighter schedule today than usual because Dr. Cohen has to catch a early flight to New York City. So we're going to have to cut things off by 4:30 exactly. So I'll try to keep my introduction as brief as possible, but I'm really happy to introduce Dr. Hassie Cohen, who has recently assumed the directorship of one of the eminent centers of aging research in the United States and arguably in the world, the Leonard Davis School of Gerontology at the University of Southern California, happens to be my alma mater, and also he's also executive director of the umbrella institution, the Andrus Gerontology Center there. He assumed all of this back in just a little over a year ago, I guess. And as I was thinking about this introduction, I was reflecting on his own history, which began in medical school in Israel at the Technion Medical School, and he specialized in pediatric endocrinology. And as I said, as I was reflecting on this, there are a number of leaders now in the field who have come into the field mid-career. In fact, sitting in the front row is one of the early leaders of the field, Dr. Ed Masro, who entered aging research at mid-career after a distinguished first half of his career in metabolic physiology. And I think also this makes me think of gerontology as sort of the melting pot of scientists, sort of like the U.S. is considered the melting pot of immigrants. So we have a lot of immigrants coming into aging research, and it enriches the field greatly, I think. It's absolutely necessary for our field because our field is everything in biology. But it's really great to see how these immigrants, scientific immigrants, are enriching our fields. And I think you'll see from Dr. Cohen's presentation a good example of that. So after leaving Israel, he went to Stanford, and he worked with Rosenfeld on IGF-1. And that has a big role, as you know, in pediatric endocrinology. But he did a lot of basic science in IGF-1, and then he moved to IGF binding protein 3, and he has distinguished himself there. And he'll tell you how that, I imagine he'll probably tell you how that led to the discovery of these mitochondrial-derived peptides, of which Humanin is the first one. And it's really quite fascinating. I think the fact that he's recently been awarded a so-called Eureka Award, and if I can remember it from the NIA, it's an R01. It stands for Exceptional Unique Research Enabling Research Acceleration, Eureka, okay, as well as a Transformational R01, which also speaks to, I think, his research. And so I'm not going to go into all the other details of his distinguished career. I think you're really going to enjoy his presentation. Thank you for coming, Hassie.
Thank you. Okay. Thank you, everybody. First of all, Jim has been a really impeccable host, and I really enjoyed meeting close to a dozen people today. Many of them I've met before. And as somebody who directs an aging research institute, I'm humbled by the tremendous brain power that the Barshop has, the intellectual depth that your institution offers, biology of aging in general. And it's a pleasure to get to know some of you. I have sort of nascent collaborations with a couple of people, and I hope that my seminar today will encourage people to reach out and expand collaborations, which is the key to research in general. And a lot of we do, and what I'll talk about in terms of mitochondrial peptides, is create an infrastructure and tools to advance scientific questions. And we can only do a very small number of the experiments to answer some of the obvious questions. And if any of you have ideas that involve the stuff that I'm going to talk about, feel free to contact me by email, and I'm always happy to offer our reagents and our assays to people who are interested. So I think that everybody interested in aging biology knows about mitochondria. They're prokaryotic, maternally inherited. They're in charge of a number of things, but mainly cellular respiration, the integration of apoptosis, and production of ROS and regulation of oxidative stress. Mitochondrial biology is well recognized to decline with age. Why that happens is still a matter of debate, but there's certainly acceleration of accumulated mutations in the mitochondrial DNA. It's the only organelle other than the nucleus that has its own DNA. And we know at the very least that certain polymorphisms or mitochondrial DNA haplotypes are associated with longevity, as well as with diseases like diabetes and cancer. And numerous diseases of aging involve mitochondrial dysfunction, particularly two that we work on that are diabetes and neurodegeneration. And there's also a strong connection between mitochondrial dysfunction and cancer. And the mitochondria, as I said, had its own chromosome that works with the bacterial code. And the mitochondrial chromosome encodes for 13 protein-coding genes, all of which are involved in the respiratory chain, as well as 2 ribosomal RNAs that create a mitochondrial bacteria-like ribosome, where these proteins are synthesized inside the mitochondria, and 22 transfer RNA genes to facilitate sort of an autonomous protein synthesis unit within the cell that's distinct from the nuclear one. And even though there's only 13 classical genes that are encoded by the mitochondria itself has, some people say, over 1,000 total proteins, almost all of which come from the nucleus, and they serve structural, functional, and importantly, signaling roles. And essentially, there are many, many proteins that are shuttled into the mitochondria and serve as signals to tell the mitochondria what to do, often related to apoptosis and respiration. But the issue of the mitochondrial cellular communication, what some people call the retrograde communication signals from the mitochondria, that remained elusive. And we had a chance together at lunch with some of the graduate students to talk about the mechanisms of retrograde communication and how previously they were thought to be primarily driven by reactive oxygen species and other chemical signals. And what I propose to you today is that the mitochondria, in fact, encodes novel mitochondrial-derived peptides, the first of which are Humanin, which serve first and foremost as a retrograde communication signal, which can then be harnessed therapeutically and diagnostically in diseases involving the mitochondria. So how did we figure out that there is such a peptide? As Jim said, my early career involved IGF-1 and its binding protein IGF-BP3 that inhibits IGF action. But back in the 90s, perhaps the first meaningful thing I've done in science was the identification of what we called IGF-independent actions of IGF-BP3, which meant that this protein had to have other protein partners. And we used both biochemical and genetic tools to identify novel BP3 partners. And we identified a couple of interesting ones. Perhaps the one that was most exciting was RXR and NUR77. And I'm not going to talk about those today because we did identify some novel genes. The first and foremost that completely transformed my own work is what we now call Humanin. And Humanin was cloned simultaneously by 3 groups. The first publication was from Keio University, a guy named Nishimoto, who moved from Harvard to be chairman of pharmacology in Keio. And he was an Alzheimer researcher. And what he said was that in Alzheimer's brains, see, this is the brain of a graduate student, and this is the brain of a full professor. And you can see that in patients with Alzheimer's, there are areas of the brain that are severely affected, a lot of dying cells, clear apoptosis, but some areas look perfectly fine. And Nishimoto hypothesized that those areas express a gene that maintains viability, and he created a library from those areas and used a survival trap where he added amyloid beta to neuronal cells and then transfected them with this library and cloned a gene that conferred protection. Right around that time, John Reed from the Burnham Institute, who is a BAX scientist, was looking for novel protein partners for BAX and also cloned Humanin. And we, trying to understand biology of BP3, cloned using yeast-2 hybrid system, Humanin as a partner for BP3. So all these groups together in the early 2000s identified this very, very novel and unique peptide, which we call Humanin. Nishimoto is the one who named it. And this peptide is encoded from the mitochondrial genome. This is the 16S ribosomal RNA region. And this is a 24 amino acid open reading frame. And we know that there's an mRNA corresponding to this, and multiple methods of fishing this out were used. We know it's produced in the mitochondria. The peptide is expressed, secreted. It's produced as a polyadenylated mRNA distinct from the 16S ribosomal RNA. And it's essentially a gene within a gene that ends up expressed and secreted. So here's the human sequence. You can see that there's various degree of conservation. Rodents have a small extension, making it a little bit larger. But essentially, it's very well conserved. The NMR structure, when you use the word NMR in two different ways today, is solved. And we've identified several distinct mRNA species derived from this region of the mitochondrial DNA that encode Humanin. And it's present in brain, testis, and several other organs. We can detect it in plasma, and for those of you who ever want to measure it, not in serum. It's associated with the clot in blood, so we need plasma, as well as seminal plasma and CSF. And its broad role is a neuroprotective, cytoprotective factor with what we call metaboloprotective effect as well. Yes?
Is it driven by the same promoter as is the gene driven by the same promoter, and is it in frame? So is it in frame with the ribosomal protein?
So the ribosomal RNA is not a protein. So it doesn't have a frame. So remember that.
Oh, I'm sorry.
So it doesn't, now it is, this mRNA is synthesized. We really have very little understanding at this time how these mRNA species are made. I'll say a few words about it later. But the issue of frame does come into play when I'm going to tell you about some novel open reading frames that we've identified elsewhere there. And those are not in frame with the larger proteins. So here's an example of, yes?
I look at, in the previous slide, you mentioned the expression of this peptide have tissue selectivity and only expressed in certain tissue. And I'm wondering, because this mitochondria peptide, mitochondria is supposed to be every tissue, so why this peptide only?
That's a great question, and leads us to the recognition that mitochondria are not uniform throughout the body. And it's already been well recognized prior to that brain mitochondria, heart mitochondria, and fat mitochondria are quite different. And people have done just standard, so proteomics, for example, and see very, very different profiles in mitochondria that come from different organs. And part of the difference between mitochondria from different organs, we've already shown, involves the expression of these mitochondrial peptides, Humanin and others. Having said that, when we identify a new mitochondrial peptide, and we do an analysis of its expression levels in various tissues. There are classical mitochondria-rich organs where there's always typically more, but it isn't completely uniform. So I think that the mitochondrial peptide signature, I don't know why I was doing this, the mitochondrial peptide signature from a particular tissue is a representation of the retrograde signaling that that tissue at any particular time has. So not only is it different from tissue to tissue, it's different from tissue to tissue with particular states. So I'm going to show you a little bit later, I don't know if I included that slide, another mitochondrial peptide that we call MOTSI that has different expression levels in different tissues, but when you fast the animals, particular tissues show dramatic changes, but not every tissue. So the ability of mitochondria in different organs to behave autonomously, essentially, and distinctly from other mitochondria is the fundamental observation that is supported and feeds into the physiological significance of this new family of peptides. I can see that it's going to be really exciting and fun, but we're not going to get through all my slides, but I guess I'll have to come back. Yes?
Just a quick follow-up. So for these tissue-specific things, do you at which level? It's transcription, processing, transport, anything?
So that's really the exciting question. And when we kind of got into this 10 years ago, there was pretty much nothing known in any detail about the regulation of mitochondrial gene expression. Now there's an emerging body of work with molecules like TFAM, SIRT3, and other genes, nuclearly encoded genes, that appear to function as transcriptional regulators of mitochondrial DNA. But this field is just in its infancy, and I think that there is, I'm looking for something to point at, am I? So I think there's going to be, first of all, distinct regulation specific to each of these mitochondrial peptides as opposed to the overall regulation of gene expression that's going to be controlled in a contextual fashion in tissues by these transcriptional regulators. And we've started now, just now, a series of experiments using TFAM knockouts, SIRT3 knockouts, SIRT5 knockout, MEFs and cells to see if we can identify how each of these transcriptional regulators affect the level of these peptides. But at this time, I have no evidence that that's the case. Better go. Arlen has a question.
This is just kind of a follow-up. So if you go back to the other slide, so are you looking at the mRNA for Humanin that comes from the ribosomal RNA? Is the ribosomal RNA produced and then it's cleaved out?
No, no. So again, the ribosomal RNA is, I guess, translated from this DNA region as a large unit with a distinct size. It's processed, it's bound to the smaller 12S unit, and there's a couple of ribosomal proteins that are specific to the mitochondria, but they're nuclear in code, and they form a little ribosome. And then there is distinct expression of mRNA completely uncoordinated with the ribosomal RNA that we don't really understand. So this is published 10 years ago, just showing the potent cytoprotective effects of Humanin on neuronal cells, and it's cytoprotective in many other systems. Here is evidence, and this is the most primitive organism that we've been able to distinctly identify Humanin, the zebrafish, and we resisted the urge to call it zebrafin. And you can see that we've been able to develop it does say zebrafin. The zebrafish Humanin is the official name, and we've been able to develop an antisense morpholino that if you inject it into zebrafish, really creates dramatic inhibition of brain development and eye development. And none of this comes from our group, but several other groups have now identified this cytokine receptor that seemed to mediate the actions of humanin. It involves the CNTF receptor subunit, the WSX IL-27 subunits, and GP130 that signals to phosphorylate STAT3, and the metabolic and neuroprotection effects of humanin work through that. And another group at the NIH showed that humanin activates ERK phosphorylation via the FPRL receptor, and the anti-inflammatory effects work with that. And we've shown that humanin intracellularly, as opposed to these extracellular receptors, work through BAX and BP3 sequestration from the mitochondria to protect cells from cell death. And so it's a 24 amino acid peptide. It's very easy to synthesize, and multiple groups. There are about 14 groups that have now published in vivo studies of humanin in rodents, and they've shown, and this is not even everything, what we call neuroprotective and cytoprotective effects in ALS and Alzheimer's models, experimental stroke, chemotherapy, toxicity in brain and testis and bone, what we call metaboloprotective and anti-inflammatory effects, and that's where our group have been very active. The reason for that is that we've shown previously that BP3 has diabetogenic effects, so we thought humanin would overcome that, and indeed we've been able to show that it both treats and prevents type 1 diabetes in NOD mice, and improves blood sugar in Zucker rats, and also in DIO models, and prevents hepatosteatosis in some models. And then there's cardioprotective and vasoprotective effects, including decreasing the size of the infarct in ischemia hyperperfusion models, a dramatic delay of atherosclerosis in ApoE knockout mice, and protection of the kidney in vivo from various insults. So here's a couple of years old paper from collaboration with our group and the Mayer group. In the ApoE knockout, developed these ugly plaques in the aorta, and you can see treatment with humanin for 16 weeks dramatically inhibits the size of the plaque. It also improves endothelial function. And a couple of published papers on how it improves metabolism in diabetic rats. It activates STAT3 phosphorylation in the hypothalamus. And you can block the action of systemic humanin by injecting ICV STAT3 inhibitors. And we like this because the relationship between Alzheimer's and diabetes might find humanin as a common link. And here is just evidence in diabetic ZDF rats. A humanin infusion IV normalizes blood sugar in about 3 hours. And ICV infusion of humanin, as well as synthetically, and particularly, we're not going to talk about it today, we developed multiple analogs of humanin that have unique selectivity in their binding partner interactions. So this is an analog that doesn't bind IGF-BP3, and it's in fact a lot more active because it appears that endogenous BP3 interferes with exogenous humanin. So this is an analog that overcomes that. Was there a question, Ashash?
Mostly based on your sequence, looks like it has a hydrophobic reach, right? It is hydrophobic reach, right?
Hydrophobic, yeah.
Looks like that based on the sequence, it looks like it's hydrophobic. So if you change some of the amino acids on the middle region on the, I think it is a leucine rich, right? Is it?
Yes.
Leucine rich, right? So if you mutate or if you change any amino acids on the leucine side, does it lose its activity?
So our group did a little bit of mutagenesis analysis. The Japanese group did a lot of that. And there's several publications sort of outlining which amino acids are essential. Because humanin has several partners and several functions, mapping structure function has to be done again for each function. So we only know some of the roles of certain residues, but you can maintain the activity humanin by, even if you lose a couple of amino acids on each side, and there are about 5 or 6 really essential amino acids. And obviously, small peptides are all the rage in diabetes these days. It's a multi-billion-dollar industry of the GLP analogs and so on. And mutagenesis of these and tweaking of these peptides is how drug companies make money and keep improving their products. Yes?
Sorry for my ignorance, but how sure you are for this mRNA from mitochondria, not actually from?
So that's a great question that people have asked. When this, the original paper by Nishimoto was rejected from Science, Nature Medicine, Nature, and Cell, and ended up in PNAS, there were still angry letters from Academy members who called it pseudoscience and didn't believe any of it. So we were very careful to identify, especially when we're now reporting novel additional mitochondrial peptides, what are the requirements? And I do have a slide about this, but what are the requirements to be able to say that a peptide or an mRNA originates from the mitochondria? So first of all, there is a model called row 0, and I'll show this if I get to it. Row 0 cells are cells in which the mitochondrial DNA has been specifically depleted. The mitochondria are still there, but they no longer synthesize new genes. And when you deplete selectively mitochondrial DNA, you lose the expression of these peptides, particularly humanin, very rapidly. Also, we can develop qPCR assays for the mRNA for humanin that's distinct from the ribosomal RNA. And we can show that's how we measure humanin expression levels. And this has to be differentiated from a phenomena called NUMTs, or some people pronounce it pneumites. NUMTs, as you know, or have heard perhaps, that the mitochondrial genome has been reintegrated many times into the nuclear genome. But the versions that are in the nuclear genome are degenerated and modified and mutated. And people have asked, could we be looking at a peptide that's actually expressed in the nucleus? So number one, we don't see it in row 0 cells. Number two, when we sequence the mRNA corresponding to this that we amplify by qPCR, it's different because the NUMT versions are a little bit different. We know for sure that the version that we cloned is absolutely from the mitochondria and not from the nucleus.
But row 0, you said it's only derived from brain and testes, but those row 0 cells
Okay, so I didn't say it's only derived from brain and testis. I say it's enriched in brain and testis. We have, half a dozen cell lines that produce humanin, and those are the ones that we made our own row 0 version. So you, in theory, can make row 0 cells from any type of cells. Some are harder than others. Mouse cells are more difficult to generate row 0 than human cells. But we have made row 0 from prostate cancer cells that make humanin, from neuroblastoma cells.
Can you get a pair of naturally occurring mutations?
So we actually worked, we haven't published this in collaboration with Doug Wallace, that he has paired cells that have varying degrees of the MELAS mutations. And we can show that humanin expression goes down when the percent of mitochondria expressing the MELAS mutation goes up, which is an interesting observation. So we have multiple lines of evidence to support that we have a mitochondrial-derived peptide here. So one of the important advances that we've made in understanding the biology of humanin is the development of immunoassays for this peptide and additional peptides. And the most important thing, I think, for this audience is the observation that humanin levels fall dramatically with age. Here's a mouse brain extract, and you can see that humanin levels falls to 1/3 between 2 and 12 months, and this is not even a really old mouse. Here are plasma levels in humans, and you can see that between middle age and near centenarians, levels drop by 30%. And the age dependency has been replicated in every study that we did, in every species that we did. In Alzheimer's patients, CSF, we can see reduction of humanin supporting a potential involvement in the disease. And interestingly, in collaboration with Amir Lerman at the Mayo Clinic, where he brings in patients for executive checkups and does endothelial functions, people who have completely normal endothelial function have humanin levels that are 2 to 3-fold higher than people who have abnormal endothelial function. No.
What about children? If you're looking at babies till puberty, is there a change in humanin levels, or is it just a...
So it's a great question, and I used to be a pediatrician. So you might think I thought about that, which I did. I'm still trying to get a nice controlled cohort of children across the ages to create a normative range. It's a lot easier to do in mice. I am going to show you a couple of pediatric samples. We've done adolescents, and there is a certain rise with puberty, and then stabilization after that. And levels are generally higher than, so they seem to be a little higher in the younger ages. That's a published figure, but we're kind of integrating multiple sample sets. So it's definitely a developmentally regulated biomarker. So we've had conversation about this today with various people, including some of the students, many of whom are still at the Barshop. But we've been trying to use many model organisms to test both the production of humanin as well as the function of humanin. And there's been a debate whether or not C. elegans actually expresses humanin, but we went ahead and created humanin transgenic worms. And you can see that compared to the control strain, at least one of the humanin transgenic strains has significant extension in lifespan. And then we went and in collaboration with Nir Barzilai, looked at his centenarian cohort. And looking at the offspring of centenarians, you can see that they're about 3 times higher humanin levels in those individuals. So both from worms all the way to humans, there is clearly some suggestion that humanin levels are associated with exceptional longevity, and maybe in a proactive etiological fashion. Another model that you're all familiar with that is associated with changes in lifespan is those models where growth hormone is altered. This is in collaboration with Andy Bartke, and this is a growth hormone transgenic mouse. And you can see the growth hormone transgenic mouse, which is bigger, short-lived, and cancer-prone, has dramatically lower humanin levels. On the other hand, this mouse, which is the Ames mouse, it has very low levels of growth hormone. It's smaller, long-lived, and cancer-free. Humanin levels are higher. And then we went and tried to look at humans. So first we looked at patients with Laron syndrome. If you don't know, they are the ones that have growth hormone receptor mutations. Walter Longo published last year that they have a very reduced incidence of cancer, although the human version, unlike the mouse version, the Laron mouse is the longest living mouse ever described. The Laron patients may not be that long-lived, but they do have low growth hormone and high humanin. Then we looked at actual children, and you can see that growth hormone deficient children have humanin levels that are even higher than regular children, and when you treat them with growth hormone, you lower the humanin levels. So if nothing else, we know that it's associated with higher humanin levels are associated with models of longevity, and several things, primarily growth hormone, regulate humanin levels systemically in mammals. So there were a number of questions that people had that I couldn't answer because I don't know regarding what might be called the mitochondrial transcriptome. And following the sequencing of the human genome, that one of the follow-up projects that was big science was sequencing the human transcriptome. And the way this was done is that 24 labs around the world were assigned chromosomes, and they were in charge of detailed analysis of the transcriptome that's derived from each chromosome. And one little lab in Australia got stuck with that insignificant job of defining the mitochondrial chromosome transcriptome. And people expected that to be a very boring project. It was a Cell paper a couple of years ago, and what they showed is a very surprising finding that not the expected 13 gene transcripts that you would think would be at high levels were found, but in fact, many, many, many different small mRNAs, some of which clearly correspond to the expression of open reading frames like Humanin. We were doing this, by the time this was published, we were doing our own transcriptomics, looking at this region here, and you can see this is where Humanin is, and you can see that there are much smaller RNAs that are expressed. And the reason this looks like this is because of the techniques involved in tagged transcriptomics. But both Humanin and other mitochondrial peptides next to it seem to be represented. And also, you see this 12S region has a couple of higher expression mRNAs that are small, corresponding to a very exciting peptide that we call MOTC, that I may or may not have a chance to tell you about today. And when you look at functions, and this is another published paper on the ability of Humanin to inhibit reactive oxygen species production, which is one mitochondrial function, right? If you add oxidized LDL to endothelial cells, you increase ROS production. Humanin blocks that. Scrambled peptide doesn't. SHLEP2 and SHLEP3 also can do that. And at that time, we also developed, this is not, I have a much better picture now that Shelley took of me kissing an NMR. But right around that time, we developed assays against SHLEP2 and SHLEP3. And I spoke with Shelley about this, and we said, let's measure. I think what you contacted me because you wanted to know what the levels of Humanin are. I forget what it was, but I said, at first I said, what are these things? And they live 30 months, not 30 years. But then, once I kind of heard more about it, I said, this is really exciting, let's do it. And what you see here is a very interesting observation, and let me walk you through this because I think it's fascinating. So in mice, you see that Humanin levels fall dramatically in plasma. From 2 months to 2 years, Humanin levels fall to less than half. SHLEP2 and SHLEP6, not shown here, SHLEP2 and SHLEP6 fall, but to a lesser degree. But Humanin falls rapidly in a short-lived species here. Then take a look at the NMR, where SHLEP2 and SHLEP6 fall pretty dramatically with age, but Humanin levels are actually maintained pretty nicely up until 14 years and only fall to 75% of baseline at 30 years. And I think what this proposes to me is the possibility that how particular species handle the decline in the expression of Humanin that's evident in plasma might have something to do with their overall biology that reflects their ability to have an extended lifespan. And I'm not proposing here that this is the reason why NMR is long-lived, but it's part of the picture that human levels remain higher. Shash?
So, but is it possible, like, are you ruling out that with age the degradation system could be different, right? So if this NMR protein, your humanin, undergo severe post-translation modifications, and they'll prevent the digestion, they'll prevent the proteolysis during the aging, whereas in the mouse on other, where they might not go through any that kind of modification that will not prevent the degradation.
It's very possible.
Your SHLEP6 declines dramatically, and in your previous slide you mentioned that SHLEP6 was pro-apoptotic.
Correct.
So could it be that in young animals where there's still development and maybe there's a greater need to get rid of cells that are improperly formed, there's more apoptosis, but in the older animals it's been switched off to prevent that?
Could very well be. And obviously, we're thinking of SHLEP6, and I don't know that I'm showing that today in the context of cancer. And we have very early data that in men with more advanced prostate cancer, there are lower SHLEP6 levels. So we think that there is some sort of programming, like you said, and the rationale that you propose now, I haven't thought about before. But for some reason, SHLEP6 fall with age, and that's perhaps the reason that there's increased incidence of cancer, or one of the reasons.
There are several databases, in particular, Mitochondria, they have this mutation associated with different human diseases. So have your data sort of make sense of those data?
We've started looking at these databases, and for me, there's two types of data that's interesting. One are germline variations in the mitochondrial DNA. And in those, we've started to identify the presence of certain polymorphisms that we're now trying to associate with changes in human levels that could be associated with progression of certain diseases. There's mitochondrial modifications that are involved in diabetes and longevity and Alzheimer's. That's one of the ones we're more interested in. Then you have data on mitochondrial DNA mutations, deletions, particularly in tumors. And those are confusing, and certainly many of them affect large parts of the mitochondria. And I'm still trying to digest those data to make sense of them. And to me, they're not going to be very useful just as stuff you download from the internet. I'd like to see them in the context of having the biological samples that we can measure the peptides in and compare them to the sequences and the mutations that are observed. But we are thinking about that, and early attempts to make sense into this are only partially successful. So here are some row zero cells that I've mentioned.
Out here at the park. Can I ask a question, please? And I'm sorry that I'm asking it now. It's a little bit beyond the slides. But can you remind me if mRNA in mitochondria are polyadenylated?
So the mRNA that encodes humanin is polyadenylated.
But does that? But within mitochondria, is the mRNA that's produced in there polyadenylated? Because if it's not, and before you said that some of these species that you see are polyadenylated, then I guess that means the mRNA is leaking out of the mitochondria.
Right. So I was trying to answer your questions, and I wasn't sure if the standard mitochondrial genes that are expressed are polyadenylated. I'll have to get back to you about that. But I'm about to tell you with this slide what might be partially an answer to your question, which is we actually believe that the translation, at least for humanin and a couple of other mitochondrial peptides, occurs in the cytoplasm, and that the mRNA not randomly leaks out of the mitochondria, but is transported from the mitochondria to the cytoplasm in a controlled fashion. And in fact, one of my postdocs is currently cloning mRNA binding proteins that may be the ones that carry humanin from the mitochondria to the cytoplasm where it gets translated. And we can obviously differentiate. The mitochondria has a different translational code than the cytoplasm. I don't know why I'm looking at the screen. I should look at the camera. The mitochondria has its own mitochondrial code, and humanin, if it's translated into mitochondria, would be a 21 amino acid peptide. If it's translated into cytoplasm, it's a 24 amino acid peptide. And early data from proteomic analysis suggests that it's a 24 amino acid peptide that it translated into cytoplasm. There was a question.
Thanks.
So it's not leakage, it's transport. And that's the life cycle: transcription in the mitochondria, translation in the cytoplasm, secretion, action on cells, and the row 0 are representative. And of course, inhibitors of mitochondrial versus cytoplasmic ribosomes is another tool demonstrating the expression occurring, the translation occurring in the cytoplasm. And this is just a row 0 analysis of all the different schleps, all of which go down when you knock out the mitochondrial DNA. And here is just showing you the diversity and the differences in the expression of each of these peptides in different tissues. And I think this is a good time to finish. I was going to tell you about a couple more peptides. And maybe I'll just jump over to mention a Andy Dillon paper showing that he can tweak mitochondria in the brain of worms, and that leads to change in mitochondrial function in the gut. And he proposed the term mitokine for a factor that's produced in mitochondria and acts elsewhere. We think they're the same factors that we have. And this is a summary slide that I hope I convinced you that the mitochondria is the source of novel peptides that serve as retrograde communication signals. They have actions on energetics and protection in the brain and the muscle, and some of them act directly on tumors to prevent their growth. They are developmentally and hormonally regulated and aging dependent, and have potential therapeutic and diagnostic significance in diabetes, atherosclerosis, Alzheimer's disease, and cancer. And in my newly renovated lab at USC, I had the opportunity to get the floor with a mitochondrial imprint. I'm very proud of that. A lot of people to thank. We have Dr. Buffenstein here, which I hope will be the first of many collaborators at your center and the folks in my lab. Thank you.